So we'll start with medical microbiology, ha? Okay. All right, so I'll be here. So if you hear the sound, kindly press 5. So, hello, dear doctors. So can you hear the sound? If you hear the sound, kindly press 5 in the chat box so I would know it. Okay, good. Sige, so let's start. That's me doing the lecture. Okay, kanina to, a while ago, siguro mga 1 hour ago, no, okay ang aming internet. Sige. Welcome to our review in medical microbiology. This is Dr. Dakanay. They call me Doc Toff. So by the
and
way,
I am part of Expert MD.
kindly take down notes kasi this is a chunk of the review, the real review of microbio. Okay? And nandito po yung mga board exam questions. So you can start taking them down. All right? So sige, let's continue. And this afternoon, I am tasked to give you an overview of medical microbiology in the physician licensure examination. All right? So, usually, medical microbiology, they consider this as a complicated subject because it covers vast topics, including generalities of microbiology and diagnostic procedures, the whole field of bacteriology, parasitology, mycology, and virology. Okay? So napakadami niya. And most of the time, ang sinasabi ng students na medyo nakalimutan na nila and they find it difficult because the questions in micro are really basic. And most of the time, sa sobrang basic nga po ng questions nila, we tend to forget it. Okay? And also, attitude ng ibang students, naaral ko na yan ng pre-med. So, madali na lang sa akin yan sa board exam. That is a big no-no. Okay? Kasi yung akala nating alam natin, hindi na pala natin siya ganun kaalam. Okay? That's why reviewing is very helpful and advisable. Okay? Especially when we talk about the board exam. So, I'm going to give you an overview of medical microbiology. Okay? And here in Expert MD, we teach the subjects, okay, based on the level of understanding of the students. Hindi kami fast-paced dito. So we always reach out sa students namin and we want everyone na ma-feel nila na sulit yung lecture hours. Okay? Promise namin na uuwi kayo after the lecture nang may naalala. Okay? That is our upbringing here in Expert MD. So let's start with micro. All right. So, this is the source of our lecture, which is the same source of the licensure examination. That would be Jawets and Tortora. Okay? And of course, I'm going to insert here yung mga mnemonics, mga tips on how you're going to remember them. Okay? Because studying is a whole different story from remembering. Okay? So don't just study, you have to remember it. Okay? Because in the board exam, I always tell my students, hindi po siya padamihan ng nabasa, hindi siya padamihan ng naaral. Padamihan siya ng naalala. Okay? So always remember that. Keep that in mind. Okay? So let's start. So this is the bacterial anatomy. And you can see here that bacteria is a prokaryotic organism. Di ba? Lahat naman tayo mag-agree. Bakit po ulit natin siya natawag na prokaryotic? Di ba? When we say prokaryotic organisms, they don't have a true nucleus. Diba? The presence of a true nucleus differentiate a prokaryote from a eukaryote. So in this case, bacteria are considered prokaryotes, right? Because they don't have a true nucleus. So as you can see here, the genetic material, let's say the DNA, is not enclosed in an organelle. Okay? Wala siyang true nucleus. Nandyan lang siya sa loob ng cytoplasm, nakakumpol lang sila. And the area in the cytoplasm kung saan sila nakakumpol, you call this the nucleoid region. So wala siyang nucleus. Ang meron sila, nucleoid. Yun yung area kung saan sila nakakumpol. As opposed to a eukaryotic organism, ang genetic material po ay naka-enclose sa isang nucleus. Is that clear? Okay. So let's go to the other organelles that are present of the cell membrane. Okay? And these are the bacteria. So when you have antibiotics that would target the cell wall or the cell membrane, it would lead to the destruction of that bacteria. Okay? Next, you have here the presence of the capsule. Although not all bacteria contain a capsule, those that contain capsule are more virulent. Later on, I'll discuss why. You also have here the presence of the ribosomes, okay, which are the sites of protein synthesis, the presence of mesosomes, and the cytoplasmic granules. Okay? And in some bacteria, they have spores, okay, but not all have them. Okay? And then outside, meron ka ditong makikitang pili and the presence of the flagellum. So later on, we'll go to them one by one. Okay? So overview yan para alam natin ang itsurahan ng bacteria. Okay? So, let's first discuss the capsule. So a capsule, okay, encloses the bacteria. And usually, it is rigid. Ngayon, kapag yung capsule po ng bacteria, yung capsule niya ay hindi rigid, meaning it's loosely attached, you don't call it capsule per se. You now call it a glycocalyx. Did you guys get that? Kasi baka when you read Tortora or when you read Jowitz, malito kayo. What is glycocalyx and capsule? Actually, they are just the same banana. It's just that pag yung capsule po ay loosely attached doon sa bacteria, okay, it is called a glycocalyx. Is that clear? Okay. So capsules, majority of them are made up of polysaccharide units. Majority, not all. So, Dok, totoo po. So, ibig sabihin may mga bacteria na hindi po polysaccharide or hindi po carbohydrate ang capsule nila? Yes. Okay? There are few exceptions. Like your Bacillus anthracis. Bacillus anthracis, the causative agent of anthrax, has a capsule. But one thing is peculiar about its capsule because the capsule of Bacillus anthracis is not made up of a polysaccharide. It is made up of peptide units, specifically D-glutamic acid. Highlight that. Remember that. Always ask in the boards. D-glutamic acid po ang capsule ng Bacillus anthracis. Okay? And that also adds up to its virulence. Is that clear? Alright. So, ano po ba ang gamit ng capsule, Doc? Bakit siya nakaka-add sa virulence? Bakit pumabangis yung mga bacteria na may capsule? Because the capsule evades phagocytosis. For what reason? Because they alter the formation of the membrane attack complex. Okay? They alter that. They play around the complement cascade. Is that clear? That's the reason why capsules evade phagocytosis. Sometimes, it can also act or has a role in attachment and adherence of bacteria to certain tissues in the human body. But this is just a minor role. Ang may major pong trabaho niyan ay ang pili or the fimbriae. Okay? And also, the presence of the capsule is one of the reasons why we have emerging antimicrobial resistance. So as you have noticed, those bacteria that are encapsulated, sila po yung madalas na magkaroon ng resistance to certain antibiotics. Okay? So remember the capsule. So as you can see here, you have here your strep pneumoniae with a very beautiful capsule that is swollen. Okay? Because of the Kellang reaction. Okay? So when we go to bacteriology, aaralin natin yan, the Kellang reaction or the capsular antigen swelling test. Okay? That's the Kellang reaction. You can also see here using the India ink method, the presence of the capsule. Ayan, streptococcal din. So you see here, di ba? Very beautiful capsule. Beautiful siya in the microscope, but once you get infected with encapsulated bacteria, okay, terrible yan. Is that clear? Why? Because they evade phagocytosis, because they alter the complement cascade. So everyone, my dear doctors, naalala pa ba ang complement cascade? Eto. Naalala pa ba siya? So we'll go there later. Ayan, naalala pa ba yan? So that is the complement cascade. Yan po yung ina-alter ng encapsulated bacteria. So we will study this in a while. I'll just show you the examples of these encapsulated bacteria. So we have a mnemonic here. You have here some nasty killers have serious capsule protection. And these are the most common organisms that are encapsulated at lumalabas sa board exam. Especially Strep pneumoniae, Neisseria meningitidis, and Salmonella typhi, and of course, Pseudomonas aeruginosa. Okay? Wala pong board exam na hindi nagtanong ng organisms na yan. So take that down. Is that clear? 2018 board exam in microbiology, set A, ang tanong po doon, the following bacteria are encapsulated except blank. O diba? Very short question, pero that's already 1 point. If you know this mnemonic, you'll get the 1 point. Okay? So it really helps na alam natin itong mga to. So, as I have been telling you, they alter the complement cascade. So, I guess all of you are familiar with this. Tama ba? First year med pa lang, dinidiscuss na sa atin yan. And first year med pa lang, ayaw na natin siyang tignan. Tama ba? Na parang, pwede ba, Doc? Huwag na natin i-discuss yan. Parang ganon lagi ang attitude natin. Kaya lang, lagi kasi itong tinatanong sa boards. Madalas siyang itanong. Number 1 in physio, number 2 in micro, number 3 in internal medicine. So lumalabas talaga siya. So don't worry. Here in Expert MD, we promise to walk you through this. Okay? So ikikwento ko sa inyo kung paano ba nangyayari itong complement cascade na to. So relax lang kayo. Huwag kayong matatakot. Okay? I got you. Okay? So let's start. So this is the complement cascade. And tatlo pong pathways yan. Classical pathway, lectin pathway, and alternative pathway. Iba-iba man silang pathway, pero isa lang ang endpoint nila. Tingin dito sa baba, ito ang endpoint nila. The formation of the membrane attack complex that would result to the lysis or destruction of the bacteria. Okay? So tandaan ha, yun ang endpoint. So yun ang goal po nitong complement cascade na ito. Is that clear? So tandaan po yan. So, umpisahan natin kay classical pathway. Everything starts here kasi ito po yung unang-unang na-discover. Pero in real life, pwede silang sabay-sabay mangyari. Okay? So, i-discuss ko muna sa inyo yung pinaka-lolo na pathway, that is the classical pathway. Dito po, sa classical pathway, it would start from the formation of the antigen-antibody complex. So, tingin po dito. Ito po si antibody, itong letter Y structure dito, and this is the antigen. So, if you still remember the structure of the antibody, di ba? Ito pong fragment sa taas, you call this the variable region, right? The variable region is the site of attachment of the antigen. So, dito magbabind si antigen. Once the antigen binds to the variable region, it would now activate the constant region of the antibody, and this constant region now will be the one responsible for signal transduction. Okay? So ulit, may antigen ka, mag-a-attach yan sa antibody in the variable region, forming now the antigen-antibody complex. Now, the antigen-antibody complex now will bind to complement 1 or C1. Is that clear? So, Doc, ano po ba yung mga complements? Kasi makikita mo, C1, C4, C2, lahat po yan complements. Complements, these are soluble proteins that are found in the serum. Okay? And these soluble proteins, when combined, can act as proteases, okay, that has effector functions. Is that clear? So, titignan natin yung mga different complements dito. So, the antigen-antibody complex now will bind to complement 1 or C1. When C1 now is activated, since it is a protease, it will now cleave C4 and C2. C4 now will be cleaved into, pag sinabing cleave, madidivide, mahahati. Si C4, madidivide siya into C4A and C4B. Ang mahalaga, si C4B. On the other hand, C2 now will also be cleaved into C2A and C2B. Ang mahalaga naman this time is C2A. Okay? So when you combine C4B and C2A, nag-combine silang dalawa, it becomes now a C3 convertase that will now cleave C3 into C3A and C3B. Okay? So we will stop here first because this is the common pathway among the three. So dito, tayo mag-stop. Mamaya, i-continue natin pababa. Okay? So the common denominator or the common pathway between the three pathways would be the formation of C3. Is that clear? Okay, so that's the classical pathway. So kung yung classical pathway po nag-i-start sa formation ng antigen-antibody complex, ang lectin pathway naman po nag-i-start siya, okay, from the detection of polysaccharides in the microbial surface, detection of these polysaccharides by your MBL. What is MBL? Your mannose binding lectin, which is again a soluble protein that is found in the serum. Okay? So therefore, when the mannose binding lectin detects mannose or other polysaccharides in the microbial surfaces, it will now attach with your mannose associated serine proteases. Okay? And the combination of your MBL and your serine proteases will now cleave C4 into C4A and C4B and C2 into C2A and C2B. So they have the same endpoint with your classical pathway. Is that clear? Which is the formation of the C4B, C2A complex or your C3 convertase that would later on cleave C3 into C3A and C3B. O, nagets yan? Di ba? Easy lang? Okay. How about the alternative pathway? Ngayon, dito naman po sa alternative pathway, it starts now from C3. Among the complements in the serum, the most abundant is C3. So maraming C3 na nakakalat sa serum. Ngayon, kapag yung C3 doon na-expose sa water, it will now be hydrolyzed. And that would now be the start of your alternative pathway. So board exam question, what is now the start of the alternative pathway? The answer is the hydrolysis of C3. Okay? So ngayon, when C3 is combined with water, it will now be hydrolyzed. The hydrolyzed form of C3 will now combine with factor B and factor D. Okay? And the combination of these three will now act as C3 convertase, which would again cleave C3 into C3A and C3B. So nakita nyo po, iba-ibang pathway sila, but they will still end up with the cleavage of C3 into C3A and C3B. Okay? So sa buhay po natin, ito lang dapat yung cleavage na dapat yung tignan at dapat yung maalala. Kasi ito yung cleavage na magdadala sa atin lahat sa pangarap natin na magkaroon ng license. Okay? So ito yung cleavage na mahalaga. Is that clear? Okay. So, dito na tayo. Let's continue. Sabi natin, C3A now will be cleaved into C3A and C3B. C3A now is important because C3A has a big role in inflammation. Tandaan, it is C3A, AAA. It has a role in inflammation. Always ask in the boards. Okay? And C3B, on the other hand, C3B, B for bida-bida. Bida-bida siya kasi iba-iba ang kanyang effector function. Number one function of C3B is opsonization. Doc, ano po ba yung opsonization? Pag sinabi po nating opsonization, si C3B po, okay, kapag dumikit siya sa isang bacteria, it kinds of highlights the bacteria. Hina-highlight niya, and therefore, pag na-highlight yung bacteria, makikita siya agad ng mga phagocytes. Na parang, uy, tol, may bacteria doon. Tara, i-phagocytose natin. Parang ganon. So, the presence of opsonization helps in our immune response. Is that clear? And that is the work of C3B. Okay? Now, since bida-bida nga siya, bukod dyan, may iba pa siyang role. C3B now, C3B can now combine with C3 convertases. Okay? Like C4B and C2A. Ito yan, it's a C3 convertase. C3BB and BB, this is a C3 convertase. So C3B now can combine with C3 convertases for it to become a C5 convertase. Ulit, C3B will combine with C3 convertase so that it will become a C5 convertase. Is that clear? So what does the C5 convertase do? C5 convertase now will cleave C5 into C5A and C5B. Si C5A, just like C3A, A for inflammation. So siya ngayon yung may role for inflammation. C5B, anong sabi ko? B for bida-bida. So si bida-bida, C5B naman po, will combine with complement C6 to C9 for it to become your membrane your membrane attack complex. Okay? So C5B, bida-bida siya, will be the one combining with complement 6 to complement 9 so that they will become your membrane attack complex. And the membrane attack complex now will yield to the lysis or destruction of the bacteria. So as you can see here, iba-iba man ng pathway, may classical that would start with the formation of antigen-antibody complex, may lectin ka that would start from the detection of the mannose-binding lectin of polysaccharide units in the microbial surface, may alternative pathway ka that would start from the hydrolysis of C3, tatlo man niyang iba-iba, isa lang ang pupuntahan, and that is the formation of your membrane attack complex that will yield to the lysis of bacteria. O, diba? So, yun lang po ang kwento ng ating complement cascade. Is that clear? And eto pong complement cascade na to, eto yung tinatakasan at nilalabanan ng encapsulated bacteria. That's the reason why immune response against encapsulated bacteria should be extra. Kailangan effortan. Okay? Kasi tumatakas sila dito. Is that clear? Alright. So, board exam pabaon ni Doc Tof. Okay? So, highlight this. Okay? You're an expert MD, kada board exam, we give you board exam pabaon. Okay? Lahat po dito. So, tignan natin. So, what are your C3 convertase? This was asked in pathology. So, it is your C4B2A and C3B-BP. C4B2A from your classical pathway, right? And your lectin pathway. C3B-BB, this is coming from naman the alternative pathway. So that's the C3 convertase. Ngayon, when you have your C3A and C5A, tandaan, sila po yung responsible for inflammation. Inflammation po yan. C3B, bida-bida, opsonization. C5B, another bida-bida, will combine with C6 to C9 to form the MAC or the membrane attack complex. Okay? And remember that your C3B, when combined with your C3 convertases, will form your C5 convertase. So just remember, pag nalito kayo between C3 convertase and C5 convertase, sino mas mataas? 3 or 5? Mas mataas si 5, kaya mas mahaba siya. So C4B2A, C3B, C3BBB, C3B. O, di ba? Pwedeng Wi-Fi password yan. O, is that clear? That's your C5 convertase. Okay? And then, the three pathways will meet at C3. So, ito po yung mga nakalap naming previous board exam questions. Okay? So, ito po yung important for you to remember. Sabi ko sa inyo, ang board exam, padamihan ng naalala. Is that clear? Alright, very good. Sige. So, ito po yung kwento. Bakit ang ating mga encapsulated bacteria like your Neisseria meningitidis, right? Your Neisseria gonorrhea and your Strep pneumoniae and Haemophilus influenzae, how do they evade phagocytosis and how come they alter the complement pathway? Okay? So remember that your meningococcal, your Neisseria meningitidis, is proven or found out already to alter the alternative pathway. Although questionable yung kanyang role in alteration of the lectin pathway. Questionable pa siya. Okay? Pero sure lang po na yung alternative pathway ang tinatamaan niya. Is that clear? Your pneumococcal naman and your HIB, Haemophilus influenzae B, alter naman the classical pathway. Okay? And then your Neisseria gonorrhea, your gonococcal infection, alter naman the MAC or the membrane attack complex. Is that clear? Alright. So in patients with immune deficiency, wherein they have defective formation of the membrane attack complex, then they are susceptible for gonococcal infection. Is that clear? In pediatrics, you will learn that. Anong specific type of immune deficiency yung magkakaroon po ng inability to form the membrane attack complex. Okay? So kapag may ganun po, prone sila into gonococcal infection. Or on the other hand, pag binaligtad naman, anong pathway or saang part ng complement cascade yung sinisira ng Neisseria gonorrhea? Bakit siya mahirap i-treat? The answer is, ito po ang kanyang ina-alter, the formation of the membrane attack complex. Is that clear? Alright. Okay, so let's go to previous board question. Okay, let's try to answer. So answerin nyo din ha, kasi ito po ay galing sa previous boards. So para makikita nyo na po yung itsurahan ng board exam. So which of the following is true regarding bacterial capsule? Diba? Sobrang basic nung tanong pero prone po yan to mistakes. So try to answer. Alright, so, okay, tingin dito. Tingin. Okay, good. So, which of the following is true regarding bacterial capsule? Letter A, always made up of a carbohydrate molecule. So, tignan natin. Tip in the board, kapag meron pong inclusion term like always or all, ganyan, pagdudahan nyo. Okay, pagdudahan nyo. Kasi baka hindi yun tama. Remember, there is no 100% in medicine. There is no absolute thing in medicine, right? So, tignan natin. Of course, except for other things. Pero most of the time, kapag may inclusion word na ganyan, like always, never, none, pagdudahan. So, just like letter A, mali yan. Okay? Because alam natin that there are few exceptions that contain a polypeptide unit in their capsule. Okay? Hindi carbohydrate. So, A is wrong. B, one type is capsid. Is this correct or not? Not. Remember that capsid is in viruses, right? It's not in bacteria. Letter B, evades immune response through bypassing formation of antigen-antibody complex. Most of the students answered this. Okay? Kasi, uy, ang dali ng board, sagutan ko siya. They answered letter C. And that is wrong. Okay? Why? Because it does not evade the formation of the antigen-antibody complex. Natutuloy yan. Okay? Ano ang na-evade nila? The complement cascade itself. Okay? Not the formation of the antigen-antibody complex. Is that clear? So the answer is letter D. It contains D-glutamic acid in Bacillus anthracis. Very correct. So letter D is the answer. Okay? Sige. Next, board exam question. Ito, recent ito. Meningococcemia is a fatal disorder affecting multiple organ systems. This is due to the evasion of Neisseria meningitidis of which component of the complement cascade? Diba? Sounds mahirap at first, but if you review, kaya siyang sagutan. What did I teach you? Pag Neisseria meningitidis, ano yung kanyang ina-alter? Very good. The alternative pathway, letter B. Okay? For classical, that's for pneumococcal and Hib, right? For the membrane attack complex, this is for gonococcal naman, your Neisseria gonorrhea. Okay? And formation of the antigen-antibody complex is never affected by the presence of capsule. Okay? So letter B is the correct answer in their boards. Alright? Kaya? Sige. Next, which of the following refers to C3 convertase? Ito, this is not from micro, this is from pathology. Okay? C3 convertase. Tignan natin. Letter A, part of the coagulation cascade? Wrong. It's part of the complement cascade. Letter B, C4B2A complex. Siya ba yan? Siya ba si C3 convertase? Yes. So that is letter B. What is letter C? What is C4B2AC3B? Mahaba. So that is your C5 convertase. And letter D, your C5B and C6 to C9 complex. This is also known as your membrane attack complex. All right. So now we go to the cell wall of the bacteria. So the presence of the cell wall gives shape and resistance to lysis by osmotic shock. Okay. Resistance to lysis by osmotic shock. And the cell wall is made up of a protein backbone, which is a peptidoglycan layer. Okay? Actually, it's a protein carbohydrate complex. So remember that all bacteria contain a cell wall except for Mycoplasma pneumoniae, right? Which is a cell wall-less bacteria. Is that clear? So, in the presence of the cell wall, so ito po yung nagko-contribute sa strength and survival of the bacteria. And we can generally classify bacteria as to being gram-positive or being gram-negative based on the characteristic of their cell wall. Is that clear? So sa board exam, mahalaga na alam po natin sino si gram-positive, sino si gram-negative. At mahalaga din na memorize natin or somehow familiar sa atin ano ang difference ng gram-positive cell wall and a gram-negative cell wall. Is that clear? Okay. Alright. So, mas maliwanag na ba? Lumipat kasi ako ng place. Okay, actually I'm here at the clinic. Tignan niyo, wala ako kasing wala akong drop light. Ay, wala akong ring light. So, meron akong drop light dito. Pwede na yan. Wala namang pasyenteng nag-chills ngayon So, I'll use it. Hiniram ko muna. So, let's continue. So, gram-positive versus gram-negative cell wall. So, tignan natin. So, for the gram-positive cell wall, kita mo dito, it has a thick peptidoglycan layer. Okay? So, tatandaan, the gram-positive and the gram-negative, both of them, they contain a peptidoglycan layer. But it's just that mas makapal ang peptidoglycan layer po in gram-positive. So it contains a thick peptidoglycan layer. Okay? Next, you also have here the presence of the periplasmic space. But remember, the thing is, the periplasmic space in gram-positive organism is thinner. Okay? Ang periplasmic space ng gram-negative ang mas makapal. Okay? So ito, compare natin siya. And then, the presence of the plasma membrane, which is a lipid bilayer, walang pinagkaiba sa gram-positive versus gram-negative. Okay? So please remember that the peptidoglycan layer, the periplasmic space, and the plasma membrane, pareho siyang meron sa gram-positive and gram-negative. Okay? So pareho silang nandoon. So the difference is that the peptidoglycan thicker in gram-positive, peptidoglycan in gram-negative is thinner. Periplasmic space, thicker in gram-negative, thinner in gram-positive. Now, ano ang peculiarities? Ano ang difference? Number one, the presence of the lipoteichoic acid. The lipoteichoic acid and the teichoic acid is present only in gram-positive. So tandaan, teichoic acid, letter T, gram-positive. Okay? Wala yan sa gram-negative. Another thing, the presence of the lipid A portion, okay? The presence of the lipid A portion or the lipopolysaccharide, take this down, is present only in gram-negative. And lipopolysaccharide is also known as your endotoxin. Is that clear? And between gram-positive and gram-negative, sino ang may endotoxin? It's the gram-negative. Okay? It's the gram-negative because of the presence of the lipopolysaccharide. Is that clear? All right. So, I will magnify this further, yung gram-negative cell wall. So, you can see here, this is the gram-negative cell wall. All right. So, you have here the inner membrane or the plasma membrane. You have here the periplasmic space, which is thick, right, in gram-negative. You have here the peptidoglycan, which is thin naman in gram-negative, di ba? Thin peptidoglycan. Then you have here the outer membrane. Now, please remember this. The outer membrane portion contains the outer portion and an inner portion. So ulit ha, yung outer membrane, dalawang portion. Outer portion of the outer membrane and the inner portion of the outer membrane. Ang mahalaga po ay ang outer portion of the outer membrane. Why? Because this is also known as the lipid A. And this is the exact site of the presence or the exact site of the location of the endotoxin. Okay? And the lipid A here is a portion of the lipopolysaccharide layer. So, Doc, bakit ganon? So, imagine again, this is the outer membrane. Outside the outer membrane, sa labas po niya, marami kang makikitang sugar molecules, which are your glucose, galactose, heptose, etc. Right? So, the combination of this sugar molecule, you call this your polysaccharide, in combination with the lipid A portion, you call it now your lipopolysaccharide layer or your LPS. Okay? So naalala nyo pa? Let's go back to the previous slide. Sabi ko sa inyo, remember the lipopolysaccharide layer or the LPS in the gram-negative because it contains the endotoxin. So, ito po yung pinalawak nating LPS layer. It contains polysaccharide and the lipid A portion. So therefore, pag tinanong kayo sa board exam, saan nakikita ang endotoxin? So the answer is specifically at the lipid A portion of the lipopolysaccharide. Is that clear? Clear po yan ha? Okay. So again, endotoxin, lipid A portion of the lipopolysaccharide. Is that clear? All right, good. Board exam question. At sagutan natin. Which of the following is true regarding the bacterial cell wall? Asan dito ang true? Okay, I'll give you 1 minute to answer. This is coming from 2019 board exam. All right. Sige, tignan. All right, so let's try. Letter A, thick peptidoglycan in gram-positive, which is absent in gram-negative. Is this correct? False. Bakit false? Because of the word absent. Remember, I pointed that out. Ang peptidoglycan present sa pareho. Yun nga lang, mataba siya sa gram-positive, manipis sa gram-negative, pero it's present. Okay, next, letter B. Periplasmic space is absent in gram-positive. False ulit yan. Why? Because the periplasmic space is present in both. Yun nga lang, sa gram-positive, it's thin. Okay? It's thin, not absent. Okay? Sa gram-negative, makapal siya ang periplasmic space. Is that clear? Alright. Letter C, LPS. Lipopolysaccharide. Specifically, the polysaccharide portion is also known as endotoxin. And mind you, most of the students in the boards answered letter C. Is letter C correct? Is it true regarding the bacterial cell wall? No, right? Why? Because it's not the polysaccharide portion. What did I point out? It's the lipid A portion of the lipopolysaccharide. That is the endotoxin. So letter C is false. So ang tinatanong which is true, so letter D, teichoic acid is present only in gram-positive. Is this true? Yes. So this is the correct answer. What did I tell you? The presence of the teichoic acid and lipoteichoic acid, letter T, right? T for positive is present only in gram-positive bacteria. Is that clear? All right. So that's it for our free subscription in microbiology. So when I meet you on the scheduled lecture in micro, we'll have more of this. All right? So thank you very much. I hope you have learned something. So thank you mga doc. Maraming salamat po.
Thank you, Doc.
Thank you, Doc.